morning, everyone. I think we can start. It's my great pleasure to have you all back after every month. It's nice to see you again and to see new faces as well. As I always mention, this is really an opportunity for the CIBM community to get together and learn and find out more and to ask questions and really to mingle. But with the circumstances, mingling is a little bit virtual, but nevertheless, it's working. So the first speaker that we have here today is Antoine Klauser, who's a research staff scientist in the CIBM MRI HUG-UNIGE Section, and he's also at the Department of Radiology and Medical Informatics as a Maître Assistant. So he will be talking to us about strategies for fast whole brain and SNR-Enhanced MR spectroscopic imaging. So the floor is yours.
So thank you for the floor. I'm nice to be given the opportunity to talk this morning. Thank you for the introduction, Pina. So today I'm gonna present to you our strategies for the fast whole brain and SNR-Enhanced MR spectroscopic imaging. I will start this presentation with a simple example of classical magnetic resonance spectroscopic imaging. You might not know exactly what I'm talking about, but don't worry, I'm gonna go into details further. But this is the acquisition parameters based on the consensus paper of 2019 that basically define the MRSI as the most classical part. This is a box in the middle of the brain where we measure metabolite signal, and we can reconstruct metabolite distribution within the box with a voxel size of typically 10 by 10 by 15 millimeter, and in an acquisition time of 13 minutes. Now, from this classical MRSI acquisition, we can go to high-end MRSI covering the whole brain in a high resolution. And now with parameters of being like 3 millimeter isotropic resolution and 80 by 80 field of view, without volume selection, we don't acquire only a box in the middle of the brain, but the entire brain, and within about the same acquisition time, 15 minutes. So my teasing question is, what's the magic with that? How come that we went in about the same acquisition time from a box in the middle of the brain in low resolution to covering the whole brain in high resolution? And that's what I hope I'm gonna present to you today clearly. And first, in different parts, I'm gonna introduce MR spectroscopy and spectroscopic imaging. Then we're gonna go into detail of acquisition, the reconstruction and the acceleration that would lead to a high resolution covering the whole brain. And eventually, I'm gonna present our result in a 2D and 3D MRSI at 3 Tesla and 7 Tesla. So first, the spectroscopy, MRS, magnetic resonance spectroscopy. Roughly, this is a method that allows to detect the difference in a resonance frequency from different components present in some body or some part of the body, like the head, for instance, in the brain. This method would allow us to distinguish water signal from lipids or metabolites. The first example I show here, MRS, single voxel measurement that was done in a voxel in the frontal part of this infant brain. You can see here. And within this voxel, we measured one spectrum with a x-axis that correspond to frequency. And you see that on this spectrum, you have different peaks. And each of these peaks can be identified as a specific metabolite, from inositol, choline, creatine, glutamate, and acetyl aspartate, with different role in the brain, from neurotransmitter, glutamate, for instance, to a neuronal marker for NAA, or choline, which is a metabolite that is present in a cell membrane turnover, but also in a neurotransmitter acetylcholine. So a single measurement like this for a volume of interest, small volume interest, really give us kind of a molecular fingerprint of this region of interest and allow, for instance, to diagnostic any disease of the metabolism in young children.
Maybe what's wrong?
Now, this volume of interest was selected typically in a slice selective way. If you are used to MRI technique, probably you know that in MRI you select you select a slice with a gradient coupled to excitation pulse. In spectroscopy, we do that, but in all three directions. Taking as example the press or steam or laser and semi-laser, almost the same sequence. You've got an excitation pulse followed by refocusing pulse, and all of them are coupled with gradient along X, Y, or Z direction in order to only eventually be left with this small volume of interest, this box in a given location. And there is a good reason for that, because most of what we are interested in this small volume of interest in single voxel spectroscopy, but also to prevent contamination from the other part of the brain. Now going from MRS, single voxel, to MRSI for spectroscopic imaging, this is almost the same. We still have a selective excitation with a volume of interest inside the brain, but now the volume of interest is generally made a bit bigger. Why to make it a bit bigger? Because now we want to add on top of this selective excitation some phase encoding in order to be able to differentiate a specific, for instance, spectrum, a resonance spectrum, and this part of the gray matter from, for instance, another spectrum in this cavity of the CSF. So on top of a selective excitation, we add some phase encoding. And this is basically what we would call classical MRSI. This makes the method rather lengthy because for each TR, actually, at each TR, we can only acquire one specific phase encoding step. So if we are in a 2D or 3D case MRSI, we need to acquire it for every single phase encoding a single TR. That makes the method quite lengthy. So now, based on that, it seems that we are quite limited in going faster in a higher resolution, because that would mean higher phase encoding step, or how can we get rid of the box without being contaminated by other part of the head. And that's what I try to I will present to you in the coming section. So a first way to accelerate the acquisition. Remember, I just told you that the acquisition time was the TR times the number of phase encoding steps that we need. If we can reduce the TR, we can already accelerate the acquisition, right? So the first way is to replace the spin echo, the press or the steam or a laser sequence with a faster acquisition scheme. That is an excite acquire sequence. Excite acquired means by the fact that the excitation pulse is right followed by the acquisition, and there is only some phase encoding taking place in between them. So in the same way that in MRI gradient echo sequence can be acquired quickly with a lower flip angle, we can apply the same principle here, means that we can lower the flip angle and shorter the TR without adding some T1 saturation in a signal. And so reducing the TR, we can probably go from a typical TR of 1,500 milliseconds to something like 500 milliseconds to 300 milliseconds. So we can already gain a factor of 4, let's say. Also, a good point is that the TE is now really short. In classical MRSI, you have a TE, an echo time of 30 to 100 milliseconds, and now we have an echo time of in order of 1 millisecond, and that is important because the metabolite signal lose amplitude with time, of course, with the echo time, and with this scheme of acquisition, we actually also gain signal. But of course, then we cannot make any VOI selection anymore. We don't have the refocusing pulse in order to select signal in space. So we have the entire slice or the entire slab that is excited, and we have of course to be careful for the T1 weighting of the signal. If we choose a flip angle that is too high and a TR too short, then we have some T1 weighting in the signal. Our implementation for a result that they're gonna present later is gonna be to minimize the TR to 500 milliseconds. And put an excitation pulse that is set to the Ernst flip angle of 35 degrees. Due to the lower flip angle, that's also a good news if we want for the excitation pulse. Due to the lower B1 needed, we can actually extend the bandwidth of the excitation pulse, and we can put it to 10 kilohertz. And eventually we optimized the timing of the phase encoding gradient that you see here between the excitation and the acquisition in order to make it as short as possible and then to keep as much signal as possible. Now that was for the acquisition scheme, the sequence point of view, but there is also the phase encoding step that we can improve. In a, let's say, classical unaccelerated MRSI, we acquire the full K-space, elliptical K-space, most of the time not really spread, it's first optimization, but let's say that's the typical way, the full elliptical K-space. And we can, using the same technique than for parallel imaging in MRI, for instance, we can skip one line over two using parallel imaging sense or GROIPA, or use more fancy undersampling pattern, like for caipirinha, for instance. At the cost of, for sense, grappa or caipirinha, measuring also some calibration data that will be needed to interpolate the point between these lines. And we'll be able to reach an acceleration factor between 2 and 4 typically. But we can also use a more fancy undersampling pattern, that we compressed sensing pattern, that is random undersampling pattern. And the good thing about this random undersampling pattern is that we can also apply some weighting function on the sampling and optimize the SNR. As you can see, we keep the full the center of the K-space fully sampled, close to DDC, and that's where most of the SNR is. So we can optimize our acquisition SNR with compressed sensing. But at the cost that, in order to compensate for this random undersampling, we need a specific reconstruction model to get our the results correct. There is also, I didn't tell you actually all the truths, that we can go faster than one phase encoding step per TR. There is spectral encoding, it's called. It's using a specific trajectory in order to encode the K-space. And within one TR, actually, we can acquire one entire line of the K-space using different fashion, from spiral to ecoplanar, concentric, radial, or rosette. This is a very effective way to accelerate the acquisition. But actually, the bandwidth necessary for such acquisition is in the order of 100 times higher than conventional MRSI. So we inherited for a lower SNR of a factor 10 at least. So in general, many average are necessary in order to reach enough SNR for a faithful for a good signal, for a resulting good signal. So it counterbalances the effectiveness of the acceleration by the number of average that we need. Now, remember, in the acquisition scheme, there is no volume selection anymore. There is no box inside the brain that we measured. We acquired the entire slice. For instance, on the left here, we would excite the entire slice of this brain, meaning all the brain, of course, that we are interested in, but also the lipid located close to the skull. And that's a problem, because the signal of the lipid, represented here by this point spread function, will leak inside the brain. The typical point spread function of a phase encoding that we reconstruct with an inverse Fourier transform would be a sinc function. And this sinc function, of course, is highest intensity at the location of the lipids by the skull, but will leak in the direction of the brain with side lobes that are, of course, maybe in order of 10 or 5% the intensity of the original location. But still, when we have a lipid signal that is order of magnitude higher than the metabolite signal that is located inside the brain, then this lipid contamination is completely overwhelming the metabolite signal. And as an example, I take this illustration from already 1996, where they show that making an image of the resonance at 2 ppm by the NAA, we see that actually this resonance is fully overlapped, is fully contaminated by the lipid signal at the skull. So we need solution for that. Some solutions are to apply filter on the K-space or to sample the K-space in a specific fashion in order to reduce the range of the PSF. If we can reduce the side lobe, the intensity of the side lobe, and make it more compact, when we are kind of resolved, because the lipid signal will stay on its location and not contaminate the brain. And one simple way is just to apply a window function on the K-space, like was done by Bogner in 2012. And in this case, the PSF, that is a sinc function, goes more into a bell-shaped function with a more compact at the cost of having an actual voxel size a bit wider, of course, but at least we don't have the side lobes and we can limit the contamination. There are also some specific, I'm not going into detail of specific strategies for sampling of the K-space, but you can read it in Kirchner et al. From 2015, for instance. There is also the solution of adding specific RF poles to saturate or inverse the signal specific to the lipids. But of course, if we add more RF poles to the sequence, probably it will mean that we need longer TR. And also these poles often are, the quality or the efficiency of these poles rely on a good shimming quality. And this is not, generally difficult to reach good shimming quality over a whole brain. So some solution can definitely exist, but this is not the easy way. There is a last solution that we have chosen for processing the lipids is post-acquisition methods. Let's say we acquire the entire data and we can process these data using a metabolite lipid orthogonality principle. Roughly, it says that a metabolite spectrum and a lipid spectrum are orthogonal. Taking the scalar product of both spectrum gives 0. Based on that property, we can actually split the original signal that contained the lipid that contaminate the metabolite and the metabolite signal, and we can differentiate both of them. And eventually we'll throw away, of course, the lipid signal that we are not interested in and only remain with the metabolite signal. And eventually, an important element for a good reconstruction would be to solve the inverse problem quickly. And also, I'm a physicist, I'm not a signal processing guy, but I think I try to explain in my way. The inverse problem, we could define it this way. In the real world, we have some metabolite we all have some metabolite distribution in our brain. And the MRI actually will measure this metabolite distribution and produce some raw data here on the right. And to go from this real world metabolite distribution to the MRSI measure the raw data, we have the forward model. This forward model normally is quite straightforward to formulate. You have the element for the B0 inhomogeneity, something for the cold sensitivity profile, and the spatial encoding scheme, for instance. And based on that, we can express what on one existing metabolite magnetization distribution, what kind of measured data we're going to get. If we want, so that's to go the forward direction from a real world distribution to measured data. But if we want to go the other way, this is the inverse problem. This is less straightforward. Generally, the problem is ill-posed, and we cannot just reconstruct, again, generally, the metabolite distribution with a single-step operator. And the solution, the precise solution required to minimize our reconstruction problem. In our case, we have chosen a specific model that was developed with Jeff Carsten and Dimitri van de Ville. This is a compressed sensing sense and low rank model. This model comprises some coil profile sensitivity in a forward model in a combination with a total generalized variation regularization on a spatial component. This should allow to fully recover from the random undersampling. The fact that we have the coil sensitivity profile that help unaliased the random aliased, let's say, the random undersampling as performed with compressed sensing will create some kind of pseudo-random aliasing in the data. And in order to resolve this pseudo-random aliasing, we need several ways. And the sense way implementing the coil sensitivity profile in the forward model is one way, and also imposing some spatial regularization is another way. And now we combine both of them. On top of that, we also assume some low rankness on the signal. I'm not going to go into details, but basically we split spatiospectral distribution into a finite number of spatial component time temporal components. And based on that, I'm going to present, based on these three let's say optimization on the acquisition, the lipid suppression, the K-space sampling, and the reconstruction. I'm going to present the result that we got. First, about the 2D whole brain high resolution at 3 Tesla. With this figure, we wanted to show the advantage of using our model reconstruction over no model reconstruction. I say that generally it's not possible to have a simple reconstruction, but in case we have a full K-space, of course, we all know that an inverse Fourier transform with a combination of the coil element should give us a rather good representation of the data. And that's what we did in a no model reconstruction. And in comparison, we compare it to the low rank TGV reconstruction. We see that the advantage of using the model reconstruction already on the resulting map quality. I mean, an obvious gain in SNR and also in the correction of the coil sensitivity profile. Looking at the spectral quality of this result at 3 location, also we see in red what was the no model recon, and in blue the model reconstruction. We see a clear improvement in SNR in this spectra. And this is reflected also if we look at the SNR map, for instance. We have probably a overall factor 4 in gain of SNR, and we have also a decrease in the Kramer-Rao lower bounds, which is an error estimate for the quantification of the metabolite. So clearly, even for a full K-space acquisition, we already got a gain of using a specific model reconstruction. On top of that, this model reconstruction allows for acceleration and compressed sensing acceleration. And that's what we want to show here with this slide. We acquired fully sampled data for 2 volunteers, 2 slides here. You see the acceleration factor 1 here and here. And we quantified metabolite from Na to GLX in both cases. And after that, we undersampled the data in a K-space in a retrospective way to simulate acceleration pattern from factor 1 to 4. So with an acquisition time that was 20 minutes for the fully acquired slice until 5 minutes for a factor 4. And that is impressive to see that the model reconstruction is able to cope with already a factor of 2 in acceleration with almost no difference. There is few difference, and this difference are more important the further we acquire, but this is almost intuitive. I mean, we acquire less point in the periphery of the K-space, and this is where high spatial frequency information is located. So of course, if we don't have these points, the model cannot make miracles, and we lose fine details data, and that's precisely what we see on this metabolite distribution. If we go higher and higher in acceleration, we lose fine details data. So that was our slice. No, I'm sorry. Based on these results, we decided to really implement that in a sequence, in actual accelerated sequence. Though we had to program the MRI sequence in order to undersample the K-space as is. A factor 2, saying that was the best compromise for a good acquisition time and a good acceleration and preservation of the signal. And for instance, we could acquire for this single slice 3 by 0.3 by 3.3 millimeter in plane resolution. And you can observe that the good contrast that are typically for instance, calling a higher concentration in the frontal white matter compared to the occipital lobe, and also the good gray matter and white matter contrast in total creatine and GLX, for instance. And again, I would like to compare that with what's the consensus acquisition for classical 2D MRSI, where we acquired a box in the middle of the brain in lower resolution and for an acquisition time of twice longer. Of course, know that there are some specialists here, and this may be a shortcut. And of course, there are some stuff that we can measure only maybe with a classical MRSI, macromolecules, this kind of thing. But if we are interested only in for instance, NA creatine choline, GLX, I mean, the fast acquisition scheme might be a good solution in order to go to whole brain and high resolution. Now, with this encouraging result in 2D, we apply this technique in 3D. So acquiring the whole brain in a 5mm isotropic resolution. Now, we made the same analysis that in 2D, acquiring a full sampled case space and doing a retrospective undersampling in order to check what would be a reasonable acceleration factor. In this case, the best compromise was a factor 3.5, and then reducing the acquisition time of 20 minutes. For a full case space, that was 70 minutes. So with a 20-minute acquisition time, we could acquire the whole brain in 3 dimensions with a 5mm isotropic resolution. And again, you can see the clear contrast in a metabolite distribution, with particularly the GLX that highlight the cortex. But also that's important to keep in mind that in this case, we have the data set containing 10,000 of voxel, and the quality of the voxel is, of course, difficult to check individually. And in order, for instance, to check the quality of some region that might suffer from bad shimming, for instance, it's important to check the SNR and full width half maximum, the peak width map. And typically, we see that in the frontal part of the brain, in the lower, close to the sinuses, we lose, of course, data quality due to the bad shimming. And this is particularly visible also on all metabolite maps. And typically on an analysis, we would need to discard this voxel based on these quality parameters. Also, checking how can we reproduce these data sets. We all know that sometimes we are just a lucky case when the measurement goes all well and we cannot reproduce the data. So we reproduce the data over 3 volunteers. And surprisingly, we had good quality. For instance, the GLX map here still preserve the good contrast of the gray matter and white matter. And also the spectral quality is particularly good. For instance, I take as an example the signal of the GLX. Now normally it's almost at the noise level. It might depend, of course, on the acquisition scheme, but in this case, at the 4 different locations that were picked onto the data set, we can clearly always see the double peak of the GLX. Also, this peak now follow clearly the pattern that we see on the distribution map. For instance, if you look at voxel located in the gray matter compared to the white matter, you see this GLX peak that are higher. Typically, the same will happen for the choline peaks as well. But this is also interesting to see that we have a really high SNR on this spectra. This is also particularly the case due to the fact that we have now a big slab that is excited. For 2D MRSI, we have typically a 10mm slice that is excited. Now we have 100mm, so 10 times higher. So as SNR, that is 10 times higher than in 2D MRSI. So we have to deal with limitation due to the difficulty in shimming, for instance, in 3D. But on the other hand, we really gain in a signal. Eventually, having this result working at 3 Tesla, we're glad to test it at the Martino Center, thanks to Ovidio Andrenesi, and the implementation of the method at 70 was kind of straightforward. The sequence is quite simple, so it's quite simple to adapt on another scanner, and the reconstruction is basically the same. So we could just reacquire it. The only difference that now we can even push further the resolution. Due to the fact that we have resonance frequency at 7 Tesla data is higher, we can even reduce the TR and keep the resolution per ppm the same. And we acquired this 2D and 3D data set at a lower flip angle than at 3 Tesla and at higher resolution. So we talk about ultra high resolution in this case on 2.5mm in plane resolution and 3.3mm isotropic resolution, and with about the same acquisition time than in 3D, say 6 minutes and 20 minutes. Again, we see more or less the same feature in a spectra and a higher SNR in a 3-dimensional data set due to the thicker excitation slab. Now in 3D, you might notice that the excitation slab is thinner. This is due to the, of course, difficulties in at 70 to acquire really a full brain with a good B0 quality, good B0 shimming. Eventually, I find it interesting to show you some preliminary results of a cohort of when we implemented the high resolution whole brain 3D MRSI at 3 Tesla at the CHUV in collaboration with the Department of Psychiatry for the ARMS cohort, where we have up to now 30 subjects where we all acquired with this method that I presented to you. And preliminary data, we decided to analyze all these subjects in a MRI standard space. That's mean that you have to warp all the metabolite map into a standard space that's supposed to represent the brain of an average population. And okay, that's a specific choice in analysis, but that's great because having all now our metabolite map on the MNI standard space, we can use all generic tools for group analysis. For instance, it could be observed, for instance, a correlation on the GLX, the glutamine level in a posterior cingulate cortex using a specific. Well, I'm not expert in this kind of clean neuroscientific group analysis, but it's a cluster-based analysis that shows that in this specific cluster we reach a high correlation of the GLX level correlated with age, and this is a well-known result of the literature. So we see that having now the metabolite distribution covering the whole brain opened the door also to new kind of analysis using registration to the MRI standard space. We can use plenty of generic tools. We used a specific voxel-based analysis, but I'm sure there's other kind of analysis as well. And also, we can easily cross-analyze this result with other MRI modalities. So to conclude, I hope that presented to you the recent technical advance that allow to really have a new approach for MRSI. The goal was not to discredit or to discard classical MRSI. Probably, definitely, we still need that, but just that we have now access to new advances that really allow to whole brain high resolution MRSI. And we developed this new acquisition reconstruction scheme that we apply to whole brain high resolution MRSI. We got some good capability for acceleration the data at 3 Tesla and 7 Tesla as well. And it was interesting to see that actually also the metabolite signal clearly benefit from the large excitation volume that we acquired. And the group, the acquisition of a group of volunteers showed good consistency. And eventually the group analysis seems to be made easy by this whole brain that could be registered on the standard space. With this, I would like, before thanking people, a future plan would be to go from Cartesian to trajectory. With the good collaboration with the Martial Center and Ovidio and Renesi, we would like to leave the Cartesian space and go into the trajectory space in order to increase the resolution and the speed. With Cartesian trajectory, we were somehow kind of limited to 20-minute acquisition for 3D. We know that's kind of strong limitation for application in clinic. So we hope that with trajectory, at least we can go factor of 2 lower. And also we would like to leave our classical reconstruction scheme and go into neuronal network, just for a practical matter. Now, typical reconstruction can take up to 24 hours for 3D data. This is due to the fact that we have to deal with huge data and huge operators, and have been optimized for several years, and it seems that we reach a hard limit. We know that with a neural network reconstruction that could be done in a matter of minutes, and of course that is crucial for to implement that straight and online and for application in clinics. And so eventually I would like to thank you, all the group of Geneva, in a Françoise Lazeras group, also Dimitri and Jeff from the signal processing core that really helped us putting on this reconstruction. I'm not a signal processing guy, but I learned a lot from them on the reconstruction. Frédéric Brouillet from the BBL at the University of Geneva, the people at the Martino Center, Vijay Indonesi and Vijaya Tapa, also Bernard Strasser that I met when he was finishing postdoc at the Martino Center, but now he's back at the Vienna University, and the Swiss National Fund for funding. Thank you very much for your attention, and that's it. Thank you.
Thank you, Antoine. Great talk.
I hope I was not too long, but
no, we have.
Okay.
Yeah, this is what the Breakfast in Science is, really to take the time to share. And
Okay.
are there any questions from the audience? Please unmute yourselves first.
So I have a very naive question, but I wanted to know, how do you know if it's reliable, the information that you sample at the border of the K-space, as you use SES reconstruction?
Yeah, so how do I know that this undersampling is not missing in the reconstruction?
Yeah, exactly. Yeah.
So, of course, I show the results at the, oops, sorry, not what I wanted to do. I show the results in comparison of in vivo data, but because I found it's a good marking. Oops, I'm doing something weird. But we did exactly the same analysis with phantom data, and we have a high resolution phantom with small tubes of few millimeter, and we can actually see that if we acquire the full K-space, we see in the reconstructed metabolite distribution where we see full details, and if we undersample the K-space retrospectively, we can still keep the same resolution in the resulting image. So of course we tested that also with synthetic simulated data.
Okay, thanks.
So
Roland, you had a question?
Well, I'm afraid it was essentially the same question that was just.
Okay.
okay.
But maybe extending on that. So have you also done, I mean, you showed this great resolution. Thanks for this great talk, by the way. Wonderful work. You showed this wonderful resolution. So if you had a patient in the clinic, so what kind of a lesion or what kind of changes you see in color contrast there would alarm you to seeing something real? Or asking, have you also done reproducibility, same acquisition on the same subject repeatedly instead of showing nice pictures for different people?
Yep, definitely. That's a really good point. On last part of the question, that's definitely some work that for future plan, definitely to have a really serious reproducibility study, scan-rescan, taking the same subject in the scanner, out and in again. And eventually, of course, it's difficult to know exactly what would be the precision. But if I show you, for instance, the acquisition that is done without model reconstruction, that's a fully sampled K-space. You see some noise here. You don't know if a single, for instance, the noise is related to a single lesion, or would that be also something related to noise? I think that's in this case, having some good phantom data and good simulation data is also the key to answer this question. Of course, in vivo, as you said, that's nice picture and it's nice illustration, but based only on in vivo data, that is not sufficient. And I think that some analysis that we did on simulation data and also on phantom data can at least ascertain that we can reach a given resolution. Now, the difference in metabolite contrasts, probably we need to have some specific phantom with difference, for instance, in metabolites concentration in order to really assess how much. Well, yeah. It's a difficult question, but I hope with some good simulation data and good phantom data, we can answer and ascertain this question, this answer. Yep.
Thank you.
Yeah, you had a question.
I also have some questions.
Yep.
I have several.
Hi.
Hey, I'm Christina.
Yep.
Nice talk, very nice talk, and
Thank you.
congratulations for the hard work done in US.
Thank you.
We are also working on something similar, but at 14 tests on the preclinical scanner.
Yep.
And I had some questions, something that is puzzling me. It's the low concentrated metabolites, because we all know that, yeah, we have changes in NA8, choline, creatine, but they are not so subtle, or how can I say, there are other metabolites which are lower concentrated, and in some diseases which change earlier and faster before you have the hole in your brain, in tumors. Yeah. you see the whole when you see the changes in this metabolite. How can I say? So it's not very useful.
Yeah.
Or can you go to low concentrated metabolites? Can you see them, or not so much? Or what can you do to see them, to see the changes?
Normally, the low concentrated metabolites are improved also by the model reconstruction. For instance, on these slides, you can see clearly that the map that are the most improved are inositol and GLX. Well, inositol in this case is not so low concentration, but GLX is, or at least a signal, not in a concentration, but the signal is lower. And most of the time it improves due to the also to the low rank assumption. Now, you have to be careful with the low rank assumption is that if you choose a rank that is too small, let's say that you choose only 8 components, then of course you will only keep signal from the main component, from the main distribution, and you might erase this small variation due to other metabolite. But if you choose something rather safe to 20 to 40 components, actually you can be sure that normally all metabolites are in. There is some empirical experiment that you can do. You can just reconstruct with a various number of components, and you just go down until you see that basically you remove noise, but you don't affect the distribution of the metabolites. So normally it should be enhanced, but of course, I mean this is preliminary, not preliminary result. Now we have this result published, but this is something that needs to be ascertained also.
Okay. It's still a nice work, even though there are still things to be done. It's very nice work. I had another question for the quantification, because the maps are very nice, the colors are nice, but you need some, everybody goes to absolute quantification. We already saw with Roland in the consensuses that there is not really an absolute quantification, there is more an unit or how do you deal with it? How do you how you make your maps uniform between one volunteer to another, and then between one center to another? You see, it's
we use institutional units, meaning that we acquire the water almost the same way than we acquire the metabolites, and then we reconstruct the water signal exactly the same way that we reconstruct the metabolite signal. So eventually we have one map of water signal that we can use for the scaling of the metabolite signal. That's the way we do that. Of course, then we don't take into account different water concentration in different tissues. So that might be also part of the contrast that is contained in the metabolite map. But this is, you see what I mean? We just in some part of the brain you might have tissue with higher concentration of water or lower, but we don't account for that. But we just reconstruct water the same way we reconstruct metabolite, and we use that for scaling in absolute.
Absolute. And the last question, the macromolecules, because you're saying that you don't really consider them. Why?
This is due to the lipid suppression method. You clearly see, I think the best way, if I go to the example of the 3D, you clearly see here that the baseline is rather flat. It looks like a long echo time spectrum almost. This is macromolecules, looks really like a lipid signal from the skull. Actually, it's almost the same shape. So basically, when you do this, the splitting of I go back to the method where I show that using the orthogonality. Then the macromolecules are not orthogonal to lipid signal. They're actually parallel to lipid signal. So they would be identified as lipid signal, and they are removed from the spectrum. So that's what I say, that in this case I'm quite sure that cannot be used to measure macromolecules.
But it's still if you eliminate them, you can still you can assume that you don't have errors because they were half there or half not there, not taken into account. So you just say that they're eliminated. It's like it's like Maudsley when he's acquiring a metabolite without the macromolecules.
Quantification. No. In this case, we're quite sure that we can see the baseline is really flat.
Thank you very much for the very nice presentation and for the answer for the questions, and hopefully we will talk more about it.
Of course, we should.
We should.